Hello everybody, welcome to our final project. Here we will explain how carbohydrates, proteins, lipids, and something else work. We are Ricardo Santibáñez, Dana Maya Chong, Natalia Romo, and Diego Esperante. We hope you enjoy it. So, to start off, we are going to explain biomolecules. Biomolecules are the basic chemical elements of all living beings. They are commonly known as chunks, and the elements are carbon for C, hydrogen for H, oxygen for O, nitrogen for N, phosphorus for P, and sulfur for S. Everything is composed by combinations of biomolecules, because by themselves they are useless. They are called biomolecules because the elements are the most predominant and versatile that compose living matter. Carbon is the most important because it has the ability to form stable covalent bonds. In this video, we're going to talk about carbohydrates, lipids, and proteins that are formed by biomolecules.
Carbohydrates, also known as sugar, they are formed by carbon, hydrogen, and oxygen, or CHO. They help us with direct energy. There are three main kinds of carbohydrates. Let's look at them. The first one is monosaccharides. They are conformed by only a small molecule with 3 to 7 carbon atoms. Some examples are glucose, that is found in the cell, fructose, found in fruit, and galactose, found in dairy.
The second type of carbohydrates are disaccharides, which are two monosaccharides linked together. An example of this is sucrose, plain old sugar, which results from the linking of glucose and fructose, two monosaccharides. Another example is the adding up of one glucose to another glucose to form maltose, which is found in beer. Finally, the third type of carbohydrates are polysaccharides. Polysaccharides are the longest and most complex carbohydrates. They are made up of at least 20 monosaccharides and have several functions. Structural, such as cellulose in plants and chitin in insects, prevention of pathogenesis in humans, as well as blood clotting, fertilization, and, within the nutrition field, the short-term storage of energy through glycogen, which is stored in the liver. Now, an important question is raised here. How are disaccharides and polysaccharides formed from such a simple thing as monosaccharides? Well, to answer that, we have to get a little bit chemical. As we mentioned before, monosaccharides are linked together to form more complex structures. This linkage is done through a process called condensation. Not the kind of condensation related to the climate, albeit, although this does take into consideration water. In biological terms, a condensation reaction is that in which you subtract water from the equation to form some kind of bond. In the case of carbohydrates, this bond is called a glycosidic bond. We take one hydroxyl group, or OH, from one monosaccharide and one hydrogen from another hydroxyl group from the second monosaccharide. When these three elements are taken away, two things happen. A glycosidic bond is formed between the remaining oxygen from the hydroxyl group of the second monosaccharide and the first monosaccharide. And also water is formed. So there you have it. Now you know how to make sugar.
This is an anabolic process, which means that requires energy. The importance of polysaccharides is that they provide energy that is later stored and then the body uses it to function properly.
Another important biomolecule is lipids, also known as fat. They are crucial for reserving energy, and they are formed by carbon, hydrogen, oxygen, and nitrogen, or CHON. They are liposoluble, so they only dissolve in other fats and not in water. The main group is triglycerides, that are found mostly in adipose tissue. Oleic acid and stearic acid are some examples. How are triglycerides formed? They are formed by 3 fatty acids plus 1 glycerol, and it also involves condensation. There are 3 water molecules removed, and an ester bond is formed.
Fatty acids are chains of carbon atoms with hydrogen. that have covalent bonds as a linkage. When we talk about saturated and unsaturated fatty acids, we refer to the number of covalent bonds that are found in each carbon. A saturated fatty acid has single bonds between each carbon, and the unsaturated fatty acid has more than one double bond. Monosaturated have only one double bond, and the polyunsaturated have more than one. What makes them different is that the saturated fatty acid is just one straight line, and the unsaturated fatty acid seems as it is broken.
We've got two energy-related biomolecules up to this point, lipids and carbohydrates. What are the differences between them, you ask? Well, in terms of energy, the main and most distinctive one is that carbohydrates are short-term energy which can be consumed at any time. That's why energy from sugars is also called direct energy, and this is also why kids go bonkers when they have one too many chocolates. Lipids, on the other hand, are a long-term energy which is stored in the adipose tissue, also known as, you guessed it, fat tissue. This is why lipids are said to provide reserve energy. But what does this mean for us? Does it mean that we must only eat sugar if we are dedicated athletes, and lipids if we expect to get through the day without much of a bump in the road? Not exactly. Yes, you should consume sugar if you want an immediate burst of energy, and yes, lipids do help you get through an uneventful day, but it's the combination of both which results in the most efficient energy production. And that's merely because you can't be completely unedged or silently sulking all the time. Carbohydrates and lipids, as everything else within the human body, is regulated by the principle of balance, homeostasis, and that principle dictates that a healthy combination of both sugars and fats makes for the best energy acquisition. Why is this? Because of cellular respiration, of course, an insanely complicated series of metabolic reactions which turns glucose and oxygen into energy. Carbohydrates yield less energy than lipids during this process, but the process itself is faster for them since they provide sugar directly. And this is why only eating sugar or only eating lipids is bad for you. Carbohydrates will mess with you in more ways than your abusive ex ever did, and lipids may be even worse, as their inefficient cellular respiration process is done through proteins called ketones, whose excess may lead to a nasty condition called ketoacidosis. And an exclusive fat diet might even turn you into a diabetic, which is rather unbelievable, but it is actually true. The lack of sugar will make your body resistant to it, meaning it will not metabolize any of the carbohydrates you consume, and the sugar in your blood will go through the roof. This is why balance is key, kids. And remember, stay away from drugs.
Proteins are also an important part of biomolecules. They are composed by amino acids, and they are linked by condensation, in which a polypeptide bond is formed. In the diagram, we can see that there are two main groups. The first one is the amino group NH2, and the second one is the carboxyl group COOH. This linkage also loses water, and a polypeptide bond is created. A protein can be presented in an elongated form, which is called fibrous protein, and the protein can also fold up when it comes this way. It is called globular. Proteins help with structure, hormones, defense, transport, and energy preservation. Those are its main uses, but it has more.
Enzymes are the perfect example of globular proteins with hormonal roles. They work as a biological catalyst. Let's look at an enzyme with more depth. An enzyme takes a substance and converts it into something that becomes useful to our bodies. The substance that the enzyme takes is called substrate, and the result is called product. The way in which it happens is a little bit complicated. First, the enzyme is moving inside the body, in which the temperature, pH, and substrate condensation are crucial. Any modifications in the environment can affect the bond between substrate and enzyme. When the enzyme is moving, the substrate links into the active site. The substrate changes into another chemical substance, and then both products separate, and the result is dissolved in the water around the enzyme.
So that's it for our project. We hope you enjoyed it, learned a lot, and I hope we get a 10. Thank you for your attention.